Medical information on this site is provided as an information resource only and is not to be used or relied on for any diagnostic or treatment purposes. This information does not create any patient-physician relationship and should not be used as a substitute for professional diagnosis and treatment. Please consult your healthcare provider before making any healthcare decisions or for guidance about a specific medical condition. Doctors Ruhoy and Kaufman shall have no liability for any damages, loss, injury, or liability whatsoever suffered as a result of your reliance on the information contained in this site. Hi, and welcome back to Unraveled. I'm David Kaufman. I'm here with
Eileen Ruhoy.
Okay, and we have a very exciting and special episode today. We have brought in a guest, Dr. Gunnar Gottschalk, who is I've been working with as a co-investigator on the Rapamycin trial, which we've alluded to in previous episodes. And we're going to spend a lot of time talking about it, and I think it's going to be really interesting. So,
welcome.
Welcome, Gunnar.
Thank you so much for having me. It's an honor to be on this podcast with you two, and I really look forward to the conversation. So thank you for
Yeah, no, it's great. I
really
appreciate you made the trip.
Yeah.
So, I would love to hear more about you and your background first.
Sure. Yeah. So I started in this disease back in 2011. I was a research assistant for Dan Peterson in Incline Village for a number of years, primarily focusing on ME/CFS patients, folks with IACCs, others. I was responsible for running the Ampligen trial for a period of time at his practice. Did a lot of work with the collaborations with the Chronic Fatigue Initiative. And then I went to graduate school in 2014. So I left, went to Rush University in Chicago and got a PhD in neuroscience, primarily with a focus in Parkinson's disease and Alzheimer's disease. And I was in a lab where we were doing a lot of repurposing of drugs. My thesis was on the neuroprotective effects of gemfibrozil lipoid in the context of a Parkinson's disease animal model. So a lot of animal modeling work, transgenic mice for the most part, and then other types of functional impairments, and that's really where we kind of ended up moving forward in this field of ME/CFS. I never really left Cimarron while I was away, but once I finished up in 2020, there was an opportunity to come back and also bring my colleague and now Chief Scientific Officer, Avik Roy, into the fold. So we started working together in 2020, primarily working on, we were interested in SARS-CoV-2. We started with testing. We and Cimarron, along with Connie Knox out in Wisconsin, who was one of our longtime collaborators, wrote one of the first emergency use authorizations for SARS-CoV-2 PCR testing before they had viral transport media issues. So we were able to kind of use what we had. We were repurposing urogenital swabs in February of 2020 to do testing.
That's early, February. Wow.
And so we got that, and then we sort of were thinking, oh, geez, are we going to get into the diagnostic world? Thank God we didn't, but we were kind of serving the community around Incline Village specifically. We tested about 10,000 people over the course of the first 6 months of the pandemic.
Wow.
Built a biobank, which is important, especially now. We transferred over to the University of Wisconsin-Milwaukee. Funny story, we needed a veterinarian for some of our animal work, and we were outside of a university setting, and so we found a local veterinarian who was willing to write our IU Cook protocol, because everything that we do is overseen for animal research. And then she ended up taking a job at UWM, University of Wisconsin-Milwaukee, and she said, well, why don't you come on over and meet the head of the chemistry department? So that was 4.5 years ago. Now Avik and I are both professors at University of Wisconsin-Milwaukee, Department of Chemistry. Our laboratory is there. We have about 7 full-time graduate students that work with us on these various projects. In 2023, we were lucky enough to have another transplant from Rush University come join us, Amnesh Barua. He is an ovarian cancer specialist, also specializing in natural killer cells, but in the context of ovarian cancer. And a lot of his work is centered around a hen-laying model of ovarian cancer, which that's the best model to study this, which is very interesting. And so he's joined us more recently, and a lot of our work is focused around these mechanistic targets. Avik is a biochemist by training, so a lot of what we do is focused on developing small compounds, but then also recognizing that a lot of these pathways, there's a lot of drugs already there in existence that can be repurposed. So we initially started working on this finding of autophagy impairment. And autophagy is just the intracellular's waste removal mechanism. So particularly, though, for depolarized or dead mitochondria, which, there's been a lot of work in this field recognizing that deficit. And so we started looking at patients from the biobank and identified folks with this elevated protein called autophagy-related gene protein 13, ATG13. This is an intracellular protein, so finding it in the blood, it's like shouldn't be there. We published the first paper and identified that this flavor of ATG13 was phosphorylated and active, and it's one of the key initial elements of the autophagy process. So without it, autophagy is aborted to a significant extent. We also found that this protein can directly stimulate reactive oxygen species, reactive nitrogen species in microglial cells, and it's small enough that it can just pass through the blood-brain barrier. So, for kind of two reasons or more, we decided, we should look at this. And so we started looking at the larger autophagy-coupled lysosomal protein degradation pathway. And then we got some funding from NIH in R21 to build a mouse model. So, chronic fatigue, IACCs, there aren't a lot of good, reliable models for this. And that is what really prevents the development of new therapeutics.
IACCs, what are you?
Infection-associated chronic conditions.
Yeah, okay.
I know, but I kind of lump them together, for better or for worse. So SARS-CoV, Long COVID, and others. But, the lack of an animal model is what's really thwarted a lot of this discovery. If you look at Parkinson's disease, Alzheimer's disease, multiple sclerosis, the animal models may not always recapitulate the exact human experience by any stretch of the imagination, but the mechanisms by which they allow scientists.
Classic animal models for each of those diseases.
Mm-hmm.
So it's interesting that you've spent time in not only sort of these more chronic illnesses that no one really seems to understand, but also time in more classic neurodegenerative disorders that there is some understanding of the pathophysiology and some of the more cellular mechanisms that have gone wrong. And so have you found any overlap in that regard?
Oh, yeah. I mean, autophagy is a classical...
It's the name of the game.
It's the name of the game with respect to gerioprotection, Parkinson's disease, Alzheimer's disease. Those are like ME/CFS, they're chronic.
Yep.
Right, but in the autophagy 13 protein that you were talking about, have they been looked at in those other diseases?
So there's about 49 other proteins that make up this sort of symphony orchestra that all works perfectly together. Other ATG proteins like ATG5, for example, is a big one. That's a serum biomarker for preclinical Alzheimer's disease. So it's one of those, again, sort of its same process. So ATG13, we're the first to kind of look at that in the context of autophagy impairment, but it's just because there was a lot of options. And that's initially what we found. But certainly, taken together, they're really convenient tests because they're serum-based. So you can just do a blood draw and, you can't biopsy the brain, obviously. Lumbar punctures are different and difficult to get. And just like in these neurodegenerative diseases, one of the big name of the game is starting treatment quite early. So even if you might have a genetic predisposition, the goal is to try to get folks early on with treatment so that you can sort of reduce the slope of disability over time. And, I think, frankly speaking, ME/CFS, as it continues on and these other IACCs, I think there's a lot of overlap with aging, particularly.
Yeah, that's my whole thing. I agree. That's how I got into rapamycin in the first place.
Right. And I remember we were at the Stony Brook conference in New York a couple of years ago, and we had been looking at this pathway, like why was this ATG13 upregulated? What was going on? And well, we found out that the mammalian target of rapamycin is the kinase that phosphorylates that particular species. So we were able to do, using the animal model, a lot of elegant experiments to show that direct link of mTOR activation. Since then, we found other evidence of mTOR activation sort of chronically in a subgroup of patients. And I think the idea of a subgroup is really important in this field because, this is really an umbrella term disease. But there certainly are patients that show elevated mTOR activation. And knowing that the mammalian target of rapamycin had a drug, rapamycin,
Right.
we were like, oh, okay, well, who's doing this? Who's willing to do this with us? And then we asked Dr. Kaufman. So
You were the chosen.
Yeah. I was the cowboy.
yeah.
So the ATG13 is a marker of activation of mTOR.
Yeah, and that's sort of our hypothesis. mTOR is driving its phosphorylation and inactivity, so it's pushing it out of its intracellular process of initiating autophagy, and it's just sort of aborting it. So yeah, but chronic mTOR is actually the target. And when we first published the paper, there was a lot of confusion from people because they're like, well, if you're saying it's high levels of ATG13, doesn't that mean high levels of autophagy? Like, no, it's phosphorylated and active, so actually it's the opposite. And then the target of that, when we built the mouse model, we built a couple different flavors of it. We first started with just a black mouse, and we made a very potent mTOR agonist just in a normal mouse. And we're really looking in the context of post-exertional malaise. That's one of the mechanisms by which, in skeletal muscle in particular, autophagy is important, right? Because you need mitochondria that are there. And so we identified when we ran these mice that we had fed this mTOR agonist on a treadmill, just like a VO2 max. They had this very significant post-exertional malaise phenotype that lasted, weeks just by feeding them this mTOR agonist.
Which was what?
Something we made. It's called
In your
Sim
kitchen?
1485. Yeah, well, we're in the Department of Chemistry at UWM, so it's pretty.
I see mad scientists.
Yeah, it's pretty cool. I think one of the awesome things about UWM and being in the chemistry department is we're kind of the outsiders. We're doing biomedical research. They're chemists. So if you have a compound, it's like magic. Chemistry is magic. And my palms still sweat when I go by the OChem labs. I'm like, oh,
Oh my gosh,
But,
I love that.
Yeah, but it's
Yeah.
something where, we can bring in sort of their expertise. And so, yeah, we just knew it was a potent mTOR agonist. That's where we initially saw this. But then we had to bring in the ATG13 story. So we built a series of transgenic genetic knockdowns of ATG13, which allowed us then to test the hypothesis sort of, independent of mTOR activation. What happens if we remove or downregulate ATG13? And those animals, too, have a very sustained and show a great model of post-exertional malaise, at least that. We've since moved on to, globally knocking down ATG13 in the mice to now tissue-specific knockdowns. So not being able to knock down ATG13 in skeletal muscle, but maintaining it elsewhere throughout the body. Same thing with knocking down ATG13 in the CNS compared to having its normal function throughout the body. So these animal models are just tools. The amount of work and discovery that's happened in neurodegenerative diseases has been based around them. And, we know that this is not a model of post-exertional malaise, but we see the features of this in an animal, and that's something very important. And again, having that mechanism is what allows us to test these different hypotheses.
Yeah, it's fantastic.
Go ahead.
I was just going to say, I think, one of the great things about working with you, and I think what informs your own work, is that you started out not in bench laboratory work. You started out clinically in Dan's clinic, seeing everybody with these horrible illnesses.
Oh,
and
yeah.
I think that...
Yeah, because Incline was where there was that.
Right.
Yeah.
So I think
Epidemic.
it really informs your work in a way that's so valuable for us, meaning clinicians and patients.
I think we kind of joke about this model of translational medicine where it's like bench to bedside. Like No, actually, it's not. It's more like bedside
Right.
to bench, back to bedside.
Totally. I completely agree. I
Yeah.
mean, we say that all the
Yeah,
time.
constantly.
I think
Yeah,
there's a disconnect in what really should be happening. And
exactly.
we like to just sort of take on,
Right.
interesting theories that at least make sense to us and at least will do no harm first
Right.
and give it a try because these patients deserve it, right? David always says, nobody wants to wait 10 years for evidence-based
Oh, yeah.
medicine,
Right.
so.
Well, and as you alluded to before, I mean, I got into it because I started reading all this geroscience, longevity, anti-aging
Right.
literature. Much of it is true literature, but a lot of it is anecdotal. A lot of it is biohacking. But it's like, it began to me to seem, wait a minute, I'm reading about my patients here. I mean, when you're 75, you have chronic inflammation and you get neurodegenerative diseases and cardiovascular cancer. But the key hallmarks of aging are related to this inflammation. Why is rapamycin working in that sphere? Wait a minute, couldn't we try it in our patients? But I didn't know any of the science. So,
Right.
this was such a fortuitous meeting when we decided to pursue this. Because
Yeah.
you had as you put it, the mechanistic answers of why this might be working.
Or at least a mechanism. And I kind of want to stress that, basic science thing, when you're doing this in these diseases, like the mechanisms still need to relate to something clinically important.
Exactly.
You're not just doing this for the sake of doing it. And that's where, again, having the background clinically in this illness, even in our current trial now, I think you were impressed that I was like calling up patients
I am
and
very
talking to
impressed.
them.
I mean, I've worked with other scientists. They don't usually get
But
down in the
like-
weeds like that.
They don't
Yeah,
want to
But
get
don't
like,
want
involved.
to.
you have to, and like that's part of the training that the students that we have at UWM, they're again kind of gifted chemistry people. But I'm like, no, you're gonna take a look at this person. You're gonna look at lab values. You're gonna look at this. You're gonna try to understand why this is important. And listening to the patient is so key because they know their bodies better than anyone else. They know if they're feeling better, and if you can again identify a subgroup, if you can help 5% of the people, that's a huge win. And Dan Peterson taught me that a long time ago with doing different experimental therapies, antivirals, things like that that the medical community would scoff at, or IVIG, other things that very difficult to get insurance coverage, etc. But there were significant. It helped a significant number of people.
Yeah, repurposing and repackaging and relabeling is a lot of what we do.
Yeah, trial and error, and trying to, put together different cause and effects in the way drugs are used, and then kind of let's just try it.
Let's try it.
As long as the risks are acceptable.
Mm-hmm.
And for us, that's our everyday clinical life.
Every day I have a conversation about a drug that I'm referring to as being used off-label. And, and it's sort of a gray area, right? It's like you have to, of course, make sure that you understand the risks, understand the mechanism of action, have a plan of surveillance with regards to laboratories and the temporal timeline that you're going to check in on those labs and what, the contact is and communication is with the patient just in case they're having a side effect. I want to know about it sooner rather than later. And so it's just it's a lot. It's energy consuming, it's labor intensive,
Totally.
But when it helps a patient, it's all worth it, right?
Yeah.
So like that's.
I mean, we had one person in this trial in particular who David, like it made my year, just seeing a change on like the KPI, like going up from being bedbound. And I honestly, when we first started this, I was like, well, there's no way 6 mgs a week is going to do a damn thing for this.
Yeah.
But then it's like, whoa, people are really feeling better. And
They are.
then, we now have some molecular data that's supporting that recovery.
Yeah, exactly.
So I want to get deep into the study results, so to speak, and how we ran it or how you ran it in the lab. But this might be a good moment to tell the other half of the ATG story.
Sure.
With the Beclin protein.
Oh yeah. So
because I think it's important. It helps people to understand it's one, the complexity of our amazing bodies, but two, this, as always in our body, the sort of yin yang feedback loops that are here, and it's relevant to this as well.
Definitely, and I think it goes back to Dr. Ruhoy's question, like,
you can call
the function.
Eileen.
What's the functional importance of ATG13? Well, there are a lot of other accepted and known biomarkers of autophagy function, Beclin 1 being being that. There's a series of others that we've looked into, but Beclin one's probably the most reliable. And, you know, what we're finding is that individuals with this elevated chronic mTOR activation actually have, you know, lower levels of Beclin, which is just a marker of autophagy at baseline, that then gets better with an mTOR inhibitor, which boosts autophagy, like rapamycin. It's the opposite story with the ATG13, right? Because the phosphorylated species is what is the kinase that's actually adding the phosphate to that ATG13 is mTOR. So at baseline, you have elevated levels of phospho-ATG13 that we can measure, and then they decrease over time. So just because we saw a decrease in this phosphorylated ATG13, you know, we really wanted to test with a known marker of autophagy function, Beclin 1, and that was where we saw sort of this inverse relationship that was beautiful. And there's a lot of other, again, surrogate markers of autophagy function, LC3A 2 and 3, WDYA. You know, there's a lot of other markers that we've included in this as well, that, again, those were discovered in the context of neurodegenerative disease because autophagy is such an important process. You know, take, for example, dopaminergic neurons and you know, the substantia nigra pars compacta that degenerate preferentially. And they do that because they have so much energetic need, right? These dopamine neurons, their extensions go from here all the way to the front of your cortex. So they're super long. So that because of that, they require a lot of ATP, a lot of energy. So any kind of change where you don't have that optimal autophagic process occurring, you know, predisposes them to die out. And then once they stop working, the striations and the projections stop providing dopamine to the frontal cortex. Well, that's where you get the trap symptoms of PD, right? It's a hypodopaminergic state. That's why dopamine replacement therapy is so good in PD for the most part, but it's not really a cure. So yeah, autophagy, you know, in the context of neurons, it's a thing. In the context of immune cells, it's everything, right? Like we've been working, and other collaborators and colleagues, you know, finding this hypometabolic state of T cells, for example, in subsets of patients. You know, we're interested in that. We've kind of also started looking at, you know, different members of the sort of innate immune system, you know, macrophages, neutrophils, et cetera, because their activity and their function is based on their ability to create energy. And energy is, you know, autophagy is a process by which energy can be created.
I think one of the key pieces here from a clinical and perspective and a patient or viewer understanding is the bioenergetic pathology, the issue, the deficits that occur, and they occur as a result ultimately of mitochondrial dysfunction, and that can be caused by a lack of autophagy or autophagy. I don't know which way we're supposed to say it.
Potato, yeah.
And can be addressed clinically and in the lab by upregulating autophagy through the ways we're going to talk about, which then helps mitochondrial function, which then increases energetics, et cetera, et cetera. And it sounds, when you say it or just realize it, it sounds obvious, but it's not obvious. But it is earth-shaking. I mean, in terms of the diseases we're talking about, CFS, Long COVID, and all the diseases of aging. It is the crucible, the crucial thing.
Yeah, it's a big target, and that's, you know, that's where, again, having a repurposed drug that you know is safe, that you know people can respond, you know, that's where it's like, oh, this is a real opportunity. Because, you know, if you had to make a novel, you know, Rapalog or something,
$100 million in 20 years.
Yeah.
You know?
And
Rapamycin is so inexpensive, and
it's been here for 50 years. I mean, it's the perfect repurposed drug in some ways.
And that's where I think too, you know, and what I kind of talk to patients and other people, it's like, well, I didn't respond. And it's like, well, we kind of know that like some people won't, but part of running these trials, it's important, you know, especially in the world where we don't have a solid diagnostic criteria or biomarker, you know, is identifying these subsets so that we can properly weight a future clinical trial, you know, and that's really the goal of our work. You know, we get a lot of guff, and we can talk more about the trial design, but yeah, it's an observational, non-placebo controlled trial.
Right.
But the reason for that is, you know, each patient is their own control. And the goal of what we're trying to do is develop a diagnostic biomarker to see who can predict therapy.
Right. I want to home in on that for a minute, because that's when you and I first started talking, that's what got me really excited was I wasn't questioning whether rapamycin worked. I had been using it for about 2 years, I think, when we talked. I didn't understand exactly why it worked, but I knew about autophagy, et cetera. But what got me very excited was the proteins that we're discussing and that you just described become a possible tool to predict responsiveness and monitor responsiveness and even adjust the dosing, since each patient may have a different individualized, personalized, customized dose. And we could, tell me if I'm wrong, but that we could adjust the dosing to get the Beclin at the level you wanted, et cetera, et cetera. And that's a game changer. It's compared to what I was doing, which is saying, let's taper up your rapamycin, we'll see how it goes.
Yeah.
Now we could measure things and predict things.
Yeah, we've been, throughout my kind of career in this, working from things like with Ampligen and other trials, finding responsive therapeutic markers has been sort of my goal. Because again, the trials fail without them, right? If you can't select the patients, and the docs are really good at selecting the patients. But when you start opening it up to a couple thousand people in enrollment, it becomes more difficult. So, certainly modulating dose, modulating sort of response to therapeutics, these are all possibilities now. And, it's not like these kits or assays didn't exist before. Like, we didn't just come up with this they've been around, but in the context of how we're using them, they've never been done that way.
You mean the assays for the biomarkers?
Mm-hmm,
but they're not commercially available.
No, not commercially. I just meant as far as, the kits that you can buy that we have access to.
Right.
One thing we did have to do in this study was we had to build a very specific, a brand new antibody to phospho-ATG13. And, we had to do that because we really wanted to make sure with clearness that the kinase activity of mTOR is what is phosphorylating on this serine residue. So we had to look at the actual structure of the antibody and then find where this phosphorylation group was occurring at, kind of redesign it. And then from there, we ended up making a new antibody with Thermo Fisher, which was a great experience, and something now that antibody will be available. And so kind of like our goal is that someone like LabCorp, if you're listening, just kidding,
Yeah, no.
Again, we're a nonprofit. We don't make any money off this, but the idea is that, this could become something more commercially available, just like the cytokine panels, right? Like, everyone thought it was crazy, 10, 15 years ago for people to do a multiplex array. Well, LabCorp does that now. I mean, all these places
I still
do
don't
it.
really even understand the purpose of those panels. Because cytokines, the different ones have different roles.
And they change so
elevated or
fast.
low, and you're
Right.
right. And they have very
that's
short half-lives, and they just change so drastically.
And they lose a lot of power that way. And then that's just because it's not a very conserved system. Everyone's immune signature is going to be different.
Exactly,
Mine, yours.
yeah.
Whereas something like autophagy, it's pretty conserved. Like, you can study it in a worm.
So I want to go back to the biomarkers for a second. I was saying, excuse me, using them could be a way to predict responsiveness. But I think we should also realize that another advantage of, bedside to bench and bench to bedside translational medicine is those people who present clinically with the criteria that fulfill the consensus criteria for ME/CFS. You have two people, and one doesn't have an elevated ATG and the other does, and that elevated ATG person responds to RAPA and this one doesn't. Why? All right? The phenotype is the same. So it gives us another way to look into other possible causes of, quote, ME/CFS, quote, long COVID, these fatiguing illnesses.
And that's why I just wanted to give you even more credit through this, because you reminded me. You were like, Gunnar, look at the non-responders. They're just as important as the responders. So, and that's because, again, there's likely a different mechanism underlying that same clinical phenotype that comes out.
Right.
That's so fascinating.
Yeah, it's very exciting.
So they have the same basically profile, but one responds and one doesn't. I mean, and we see this clinically all the time.
Yeah, sure.
I mean, sometimes rapamycin is like magical for some patients, and then sometimes it doesn't have any effect at all, and then sometimes it has adverse effect. So let's talk about rapamycin.
Yeah.
Though. So, when I first, I just like David, I use it regularly in my chronically ill patients, ME/CFS and long COVID, and I definitely have a good percentage, though not the majority, of responders, and I just, it's so interesting to me because initially my familiarity with rapamycin was how I used it in neurology for diseases like tuberous sclerosis and
Right.
those those kinds of abnormal growth disorders. And so I was just sort of fascinated when I read about how other uses for rapamycin just based on its mechanism of action. And it made so much sense to me that it could be useful in this population just from the literature.
Yeah. I mean, like
And you already sort of explained like why you clearly thought, rapamycin is an easy answer, at least to first start studying, just from the work and autophagy that you had done.
Yeah, and I think just mTOR is such a big target because it is just a central growth control.
Well, it's so conserved. It's in every living organism on the planet.
Yeah, but I think to kind of answer your question both about the responder status is, there's a couple of very important things to remember, like, the metabolism of this drug is through like CYP43A, cytochrome P450. That's a known drug thing that, the docs have to regularly check for a series of things. So that's a big one. We probably don't have the power right this second in our first phase to see it, but I will just say, there appears to be some sex differences, which, is with females and males, specifically males seeming to respond a little bit more than females. Again, we don't have a big enough cohort to really understand why that is, but there's definitely something there. I also think that, there are different ways, particularly clinical markers that are accepted, like cholesterol levels, et cetera, that might inform dosing schedules because, just because you have someone on the same dose, doesn't mean that their body's going to, change how that's happening. So, and then finally, we found very clearly that there's variations in the different generic formulations of the drug, which is like, that was scary to me. It wasn't scary. It was just not something that I expected.
Realized that the variation in bioavailability significantly can affect the outcome that we're looking at. So I want to re-emphasize what you just said. So we were talking about non-responders. Well, they may not be non-responders. Right. They just may be on the wrong dose.
Exactly.
Right? And that's, again, why the biomarkers can be so good. So maybe the patient who's clearly got ME/CFS, meets all the criteria, has terrible PEM, but their Beclin doesn't rise with rapamycin, well, maybe they need 15 milligrams.
Exactly.
Or 25
right,
milligrams.
And maybe we need to review their list of medications.
Yes, exactly, and all the interactions and the pharmacogenetics of it. I mean, it's such a wide open issue, and it's so important to realize that non-responding doesn't mean, the drug didn't work for you. It might mean that we just need a better idea of dosing.
Yeah.
Right, and again, that's where this next, the next phase of the study, we're trying to address those critical concerns, partnering with a group that does a lot of work, Ageless RX. They've done double-blinded randomized placebo-controlled trials. They have a lot of what I would say is very impressive data on the bioavailability issue of the different generic formulations versus the compounded generic formulation. They also, again, have access to just sort of centralized labs. I mean, the fact that we can now, in this next phase, people are getting drugs shipped to their door, I think is like the coolest thing ever. Like,
and they're getting the same drug. Everybody gets the same
exactly.
product.
And that's kind of
it standardizes everything.
And and then that will help us immensely. We're also doing CYP43A, cytochrome P450 genotyping, and that will help us understand, slow versus fast metabolizers potentially.
Plus, we're adding in sirolimus levels,
of course. Can we just take a step back? Because I know you guys have been
it's hard to hold us, control us. We're too excited.
Too excited, yeah.
Can you describe the study design sort of from the beginning?
So, this is a prospective observational clinical trial that's not controlled. So basically patients, we take a blood sample and clinical data time point at baseline, about 14 days plus or minus, before they start the drug. And then we take a second time point after a ramp dose increase. In the first phase, we did a 6-week ramp dose increase. So patients started on 1 milligrams and then added weekly rapamycin by 1 milligram up to 6 milligrams. That got us to about 36 days. That's where we took our T1 time point. And then we had second collections and third collections, T2 and T3, 30 days after that, respectively. So we got 2 time points of patients on the full drug plus the ramp. And I think one of the interesting things that we found is I really wanted to see we didn't quite really know the dose. I mean, and Dr. Kaufman, made a good guess about that, and and it was a very conservative approach, I think. But again, we wanted to see if we could see any quick changes in our molecular assays, which surprisingly we did. By the time patients got to T1, we were seeing significant fold increases in Beclin, fold decreases in phospho-ATG13 that were continued out. But then it came to the point after the 60-day time point, and the patients were like, I want to keep going. And we didn't really think that we should increase the time points. Well, made an IRB approval, protocol amendment, and so now we're collecting further out. So in this next phase, we're doing the same type of ramped approach. We have reduced the ramp period from 6 weeks to 3 weeks, just given that we didn't see a lot of chemical sensitivity issues. That was sort of a known thing that we were expecting in this patient population. So the ramp is shorter. We're on the same bioavailability equivalent of drug from ageless. We're using a compounded generic this time rather than whatever Walgreens and whatever was hawking that were all different. Again, the differences in the drug, we wanted to standardize that. And we're collecting, the first 4 time points baseline through T3, and then we've extended that sample collection out for 1.5 years, where we're collecting those then samples and clinical data every 4 months. So
let me just say something about the dosing. I'm not sure our listeners, I don't want to make it sound like I just came up with 6 milligrams. There was a rationale there, okay? And it's important because it goes to the science, okay? So something like 15 or 20 years ago, Joan Manick, who's a MD PhD, I think, did a trial using rapamycin because of their interest in geroscience and its potential role in immune competence, okay? Rapamycin, we know, and I think our listeners know, in its usual dose is given 6 to 8 times higher than we use to suppress the immune system so you can have a transplanted kidney, lung, heart, whatever. Okay. So it's another drug, a little like naltrexone with this hormesis, where when you change the dose, you get a completely different response. So she did a trial with people over the age of 65. I'm not going to say elderly because I'm biased. Over the age of 65 during flu season in Australia, I think. Okay, 2 groups. And I think she used 3 different dose levels and a placebo. And what she learned in that trial, and it's really a landmark trial, was that at the dose we're talking about, 6 milligrams a week, there was no difference in adverse events compared to the placebo group. The 6 milligram a week had a much greater response to flu vaccine, which was one of their markers for immune enhancement. But what they didn't expect is that after the trial ended, in terms of giving the drug, I think it was an 8-week trial, but it might have been 12, I'm not sure. Those patients on the 6 milligram a week dose for the next year had a significant decrease in all respiratory illnesses compared to placebo. So that's where the 6 milligram dose comes from. And the same limitations apply. She used a single product because it was her drug company, but I don't think they did, 450 analyses and things like that. So, but that it does come from a real trial, placebo-controlled trial. That's the key piece.
Yeah, and I think the weekly dosing is it's great, and you can get away with it because the half-life of the drug is long. And
right.
That's sort of the key. And in this, in this next phase, in addition to looking at the genetic predisposition markers of drug metabolism, we're actually going to be doing quant sirolimus peak and trough levels. So we've designed it so that the Cmax of the drug is about 6.5 hours after you take it. Half-life is 66.5 hours. So patients, if they started on a Monday morning, take it with food. In the afternoon, the mobile phlebotomist can come to their house, take a blood draw for the peak levels, and then repeat that process on Wednesday of that same week so that we can get a good understanding of, and combine that with the genetic predisposition testing and see what we can see.
So again, to underline for listeners, if somebody is entered into the trial, a patient, whether it's my patient, your patient, or anyone else, that timing is key. All right.
Yes.
I mean, this is a moment where, we could learn so much from this trial about science and this disease, and we need our patients who are in the trial to cooperate with that timing issue, which is not a huge demand, but it's It's a little
Oh,
a little
it's.
bit of a
I know it can be
pressure.
tricky.
I would
Yeah.
say,
Yeah.
and I would say, yeah, we've been like, oh, decentralized trials, and oh, like we're making this easy. Like it's still hard
It's still hard.
Right.
to manage that, and that's also like,
And you want it to be unassailable, right? Especially in this
Right, right.
controversial
yeah,
disease
But we can,
space.
if people follow the rules,
yeah,
Right.
we can learn so much because
Absolutely.
nobody's done this time sirolimus levels.
yes,
Nobody's done single, except for the Joan Manic trial, the single product, so to speak.
Mhm.
exactly.
So this could be huge.
And I
Yeah, and
think
I'm glad you explained those trials because I'm regularly, because of course Google can be an enemy.
scary, yeah,
Right.
yeah.
So patients will Google rapamycin and it'll immunosuppressive,
Always.
used for cancer, used for transplant.
Yeah.
So I'm regularly explaining to patients how low this dose is and how it does not have that kind of
Right.
effect.
And that's a problem with their PCPs, their primary care docs and other physicians. They're afraid of it
Yeah.
because for 50 years it's been given to transplants. So,
Right.
Like,
know,
why do
often
you need
say
this?
things- I often say things like, we use other drugs these days for transplant and cancer patients because we've got way more toxic stuff to give them.
Yeah.
know?
Yeah.
So I'm glad that you explained
Yeah.
those trials.
And we
which
really didn't. And David, you can talk more about this, but I mean, I looked at the AEs, the adverse events from the phase 1, and honestly, we didn't see any change in
Yeah.
like your propensity to get a SARS-CoV-2 infection or a flu or anything.
Right.
So
Right.
I
I
feel
have not
really
seen it at all
confident. I feel really
clinically.
confident that the immunosuppression thing is not a
not a-
deal.
it's
in
not
Not
an
this
even
issue.
like the known sort of non-dangerous side effects, but like in one patient, I saw a lipid panel go like out of way high
Yeah.
within like 2 months time, and so I felt it was clearly related to the drug, so we stopped it. But in general, I haven't seen any mouth sores.
Yeah.
I used to give dexamethasone mouthwash to patients like with tuberous sclerosis, and I was doing higher
Yeah.
dosages. But I haven't seen any of those. And other than that one patient, I haven't seen significant changes in lipid levels, and I haven't seen significant changes in glucose levels.
Yeah, I mean,
So-
we took all of the clinicians' data, from phase 1 and just because we were interested in lipid values and no
Very,
difference.
very little changes.
And if anything, it seems, and it could just be America. I don't know. I don't see patients like you guys do, but like a lot of people in America have kind of elevated triglycerides and stuff. But so they're not even outside of the range of what would normally be considered abnormal.
Right.
And again, like other markers of related things like hemoglobin A1C or others, yeah, they're right in line. And that's again where, I think the lipid peroxification pathway, I think that's really important in this disease, but I don't think we have the tools available in like LabCorp or Quest to really understand that yet. So, but I do think as far as like a response to drug, like if someone's LDL shoots up or triglyceride shoots up, yeah, you could back down on the dose, and
Right.
probably that's
Right.
something.
That's why we follow it. That
Exactly.
until you came along with the biomarkers, for me that was how I adjusted dosing. If their
Right.
A1C went up
Right.
and if their lipids went up, I would lower the dose. What I haven't had the guts to do, but I toyed with it, is if it doesn't go up, maybe I should be increasing the dose. But
And we're
I'm a
hoping
little
that
nervous about that.
maybe if we see someone, for example, whose Beclin went up a half a fold instead of threefold, maybe we can say that this study, Dr. Kaufman,
Exactly.
you tell them to go up.
Exactly. That's my goal here.
Okay.
Cool.
Cool.
And actually, since you bring up Beclin, let me add one more thing. You and I were going over the paper, editing for submission and looking at the data. And one of the things that I saw that I didn't realize that you confirmed is that the Beclin increase is not binary. It's not like overnight. You take your dose and
Really
it doesn't happen the minute you inhibit mTOR. And at month 2 to 3 is when you start to see it max out at the higher level. So it's important clinically to be able to tell patients, don't give up after 1 month or 2
Yes.
months. This is a therapeutic that may take 3 to 6 months for you to see the ultimate ongoing benefits of it.
Exactly. And it's the same thing with people reporting how they're feeling, I think. But that's critical. I mean, that piece of data showing that really it's T2, T3, and probably beyond. I was kind of thinking this was going to be more of a supportive type therapy where we would see a quick on-off switch.
Yeah, that's what I expect.
But it's obviously that's not the case.
Yeah.
And then, we have some future ideas as to why that may be, particularly with the immune cells, like class switching, a lot of cool things that are coming out of the samples. Like this is a great study design because we took a lot of samples. And so when you build a biobank from a trial like this, you can like have someone validate your work. Very important. You can have the baseline materials that you need to do a lot of different assays, even just taking PBMCs, like, that's something that we can utilize, to further understand mechanisms of how this works. And one of the next things we're actually using is some of the leftover plasma and serum, the aliquots that we made. We made thousands of aliquots on each person, but we have a cell culture model that we can douse the serum on these cells, and we can see how those cells change based on the drug status of the patient, whether they're baseline or on the drug. And, there's so much information in just that fluid itself. And because we have an intervention, we know that there's a known change. There's a lot of people that are doing work in this field. For example, VO2 max testing, like pre and post testing, what's the difference? Large shotgun metabolomics, proteomics stuff. But here, what I love about this is we're doing an intervention that's safe and helps people. Like a 2-day CPET test is like a killer for these people. Like,
oh my gosh,
why would you, like, I just feel like there's a lot of other ways to design, platform trials like this where you are looking at an intervention and looking at a mechanism. And, I'm on a committee with NIH looking at, Long COVID Recover Committee, looking at different drugs and utilizations. And, I think these decentralized trials are good when you have a drug that's available, that's safe, and that's oral, right? IVIG is going to be tougher to do an at-home clinical trial with, but there's these opportunities now where you can study treatments and where the patients are their own control. And again, that will get you to the subgroup question. So then you have a lot of confidence if NIAID or CDMRP, they want to fund a double-blinded, randomized, placebo-controlled trial of something like this, you can say, well, we're going to add this to the diagnostic criteria, and we're going to add this to the inclusion-exclusion criteria. So when you go to randomize something, okay, we've got something here.
Right. Let's talk for a minute more about the decentralized trial concept because I'm biased and I think it's great, so important. I mean, if this were a typical trial, like pick a drug, it doesn't matter. It would involve, 3 months. It would involve probably 5 office visits and a huge amount of paper. And you'd probably have to hire a research organization. And the cost, I mean, we're desperate for funding.
Yes.
But nevertheless, the cost, if we did it the old way, would be tenfold.
Oh,
at minimum tenfold.
At least.
And so it allows people like you and people like us who don't have access to or easy access to the usual funding sources to still do some real discovery work and translational medicine work. And it's the same thing we did with the oxaloacetate trials.
Exactly.
And it's key, and it can be done. I mean, obviously, there's certain things we can't. Like you said, IVIG, you can't decentralize it.
And I think, even for ideal, since we're writing this manuscript, it's like, what are the limitations? Oh, there's a lot. But, I will say, decentralized trials, like they are going to be the future of this disease, I think, particularly with repurposed therapeutics because of the cost issue. I also think that now with telemedicine, how it works and set up, it's very possible to do that. I will say that probably for a bigger, more expensive trial, for example, if you're using a JAK-STAT inhibitor or if you're using something like IVIG, you'd probably wouldn't want to try to do a decentralized trial. You'd want people in the office. You'd want to have your pause.
Well,
on
I think as the adverse effect profile rises, then you more and more.
But this was a perfect opportunity, and like on this committee, they were like, mobile phlebotomy is so expensive. It's like, no, it's not. There's a lot of burned out nurses that quit healthcare, after the pandemic, and you find these people and you develop a relationship with them, and in a lot of cases, it's a lot easier for the patients. I mean, try making an appointment, even if you have a Quest or LabCorp requisition. It's impossible. Yeah. So there's some things, because we're trying to develop a diagnostic, we were very cognizant as to the process of how you would do that, and part of that is shipping samples, right? Like, if it's impossible for a mobile phlebotomist to send you a good sample for your stuff, then it ain't a good diagnostic. Okay, so that's why we've been focusing on this, overnight shipping and handling through FedEx, which is great when there isn't a natural disaster, but
when
that
weather
happens.
permits.
But but it provides us a way. So like, we're not shipping these things on ice packs. Like I I refuse to use ice. Okay, like we need to think better about that, because yeah, of course that will jack the price up. I mean, the fact that we're shipping these kits out in styrofoam boxes, that doesn't sound like a big difference, but if you start using big insulated boxes with dry ice, all of a sudden your shipping and handling goes from, $30 a pop
Well,
to
and also
250
Yeah, I know.
per sample, and it's
and
like
I do
increases
a lot of
sample
shipping with
failure
samples.
because a
100%.
percentage of people, not intentionally, whatever, the ice melts, it doesn't stay cold, and when you get the sample, it's no good.
And there's great new products like Stem Cell. I'll shout them out. We use their magnetic bead separation kit for PBMCs, and that means you can take a PBMC, you can have millions of live PBMCs, which is super important for downstream applications like seahorse, where you're doing functional assays. You can take millions of those samples in a K2 EDTA purple top tube, 8 mLs. It can sit on the bench or in FedEx for 3 days, unrefrigerated, and you get perfect PBMCs.
Yeah, it's so key.
And that's where, again,
That's
leveraging
really good
those
information.
technologies. Oh yeah,
I have such problems shipping things on ice. Even, yeah.
I
'cause
feel I
you-
tell
you
people
take
like
a-
I got a PhD in FedEx. I know how to do it. And Granger, I like the two companies. I'm just like,
That's
these
good.
are
I like
great
that.
companies.
Oh my God, you have to tell me those secrets, because we really do. And this is not the topic of our podcast.
So the other,
I need to know that
thing
stuff.
in the Decentralized that I love is I remember doing all the HIV research I did back in the dark days, and you drown in paper. I mean, I remember these
Oh yeah.
data collection books. Each one was like this
Yeah.
big.
Yep.
For one patient, right? Now we do it all in REDCap. It's
Yep.
all done online.
Yeah.
It's just keyboard. And then
Their
it's certainly
phones.
one thing our patients can do is keyboard on the computer or on their phone. So it's so patient friendly on top of everything else.
In this round, and what I actually was able to do retroactively, which we're going to include in the manuscript now, is even things like collecting step count. Okay, like I know it's not perfect, but that's another thing where patients, if you have an iPhone or an Android, it'll give you like your monthly step count, something like that. But certainly REDCap is a game changer for us. That's run by Dr. Maureen Hansen's lab. We have great support. We were going through the manuscript and we had some deciding about a cohort of group, these responders, partial responders, etc. Well, I had a conversation with their data guy at Cornell, and he said, I'll just build a machine learning model. And I was like, wait, what? Okay, this kid's like 22, Brian. He's like the smartest guy in the room everywhere he goes. He's starting medical school in Tufts in a couple of months, so I'm just like, please help me. But no, just over the past couple of days, and I'll show it to you, he was able to build a machine learning model that predicts responders based on questionnaires, and then integrate that with another machine learning model that integrates with our biomarkers. So we have two ways of skinning
And what
this
about
cat.
just even, like you mentioned, steps is one, but other data points that with these gadgets these days, and you can stream them, like just even vital signs, for example. Are you incorporating those?
Yeah, we've got a group of patients. We've got about 15 folks that are on these Garmin watches. I like that because they have a really good VO2 max estimator. There's another group based out of Minnesota called Moxie, and it's measuring oxygen consumption in muscle tools and muscle tissues. So you wear this like sleeve on your leg, and it's made for basically.
That sounds really interesting.
Yeah, and it measures like oxygen consumption rate too. So you get a really interesting sense, and it was designed for elite athletes and weekend warriors who want to do triathlons or whatever.
There's also this external device, and maybe this is sort of too far out there for this particular phase, but there's now an external device that will measure intracranial pressure.
Yeah.
And in these patients, there's very commonly a pressure disorder of some sort, usually hypertension, but sometimes intracranial hypotension. And so it's hard to obviously measure unless you're going to do a lumbar puncture or do bolt monitoring. But now I think it's called Brain for Care.
Yeah, I haven't seen that. Wow.
Yeah, and
That's cool.
It looks like a headphone, but it's not on the ears. It's around the temples, basically. And it's some sort of pressure transducer. And so it measures intracranial pressure. Anyway, I just think it's an interesting device, and I just wonder how I can incorporate it into.
Along with that, I didn't know about that one, but it makes me think that the Lumia device, the one that does.
I don't think that's all that reliable because it's just measuring the flow of arteries that I don't think are representative of cerebral blood flow.
Oh, okay.
So I don't know.
They're gonna try this Moxie thing. I contacted the guy, and I think he gave us 3 free kits.
I'll try it.
Yeah, no, I already wore it. My wife wore it, so we're usually the controls because we're.
Yeah.
But yeah, it's cool because it tells you again, really what we're interested in is oxygenation and muscle activity, and so it's sort of an infrared light directly into that part. But again, I will say this, the wearables are great. They're expensive. What we've shown and what we know in this disease population is like the questionnaires are actually pretty good. So I know we want to move forward from that, but that's where I keep like telling the patients, like, please read the questionnaires carefully because they kind of change. Like we're talking about the last 2 weeks or since this time point, but those are generally good predictors. So it's another way you can just keep the cost down. And I mean, this model of utilizing REDCap, like I called up Maureen Hansen. She said, sure, you can use REDCap. And that's something where sharing resources like that is what we have to have. I think Cimarron, like our laboratory, we're in a shared space at the university. Our rent is very low, but that provides the ability for us to get access to core facilities to do things like that. And so you have to hustle and grind because there's not a lot of money in this. It doesn't look like there's going to be a lot of money in this moving forward, which is a bummer. But at the same time, even small things like step count, like a lot of researchers, they just kind of, because you get so caught up in what you're doing, it's so easy to like kind of armchair quarterback something after the fact, but it's really hard in the process to see that. So, and it's a lot of effort to go back and get that information after it's done. So if there's neat ways to integrate it into everyday lives, like the wearables, I'm all for it. The apps, like Visible is a great app. It started in the UK. Those are all things that are super important. And there is a difference though between kind of research grade, like ready for prime time devices versus kind of more exploratory things. And, that's just because they're validated devices. Like FDA validates specific devices, just like they control certain drugs and stuff like that. So, but again, if I see someone's estimated VO2 max go up 20 points, I'm like, you can't train that, right? Like, you're for the most part, normal, healthy people, like, yeah, your VO2 max is your VO2 max to some extent.
Even more significant because the way my VO2 max might go up is because I work out.
Right.
But our patients cannot work out.
Exactly.
It's one of their cruel catch-22s for our patients. They cannot do exercise, which is a great therapeutic.
Of course.
So if we could show that rapamycin raised their VO2 max, that's huge.
That is
groundbreaking.
That's amazing.
Yeah. And, but again, the decentralized trial, I think it's going to be, the way things move forward. I don't think people need to, I don't think you just need to go into these brick and mortar facilities to do a lot of this stuff.
I mean, there are several rapamycin trials going on now, which is, I'm sure, frustrating in some regard because I think money is sort of dispersed in terms of supporting these trials. But how is your trial? How is it different than? Are you even aware?
Oh
of
yeah,
the trials?
no,
sure,
yeah.
I figured you were.
Yeah.
But how is it different? What's the distinction about what you're doing in your lab versus some of these other trials?
Yeah, I mean, I think as David and I talked about it, we're kind of psyched that, the greatest form of what's that? Whatever, blah blah blah, blah blah blah. You know what I mean?
Impersonation.
Yeah, whatever that is saying.
Flattery.
But I think at the end of the day, we have this sort of mechanistic target, and in 2022 when we first published this article about elevated phospho-ATG13, people are like, whatever. Now we start talking about the context of autophagy, and like it gets cited now like all the time, and that's how it happens. Like science, you're usually going to be out alone for a long time before someone starts looking at it and validating it. We are looking to develop a predictive diagnostic. That's honestly the reason why I'm interested in doing this. That's where we don't need the double-blinded randomized placebo-controlled trial. I do see great value in a double-blinded randomized placebo-controlled trial, but I know that it will be expensive to run. And I also know that it might very well be duplicative, knowing that in the geroprotective world, for example, millions of people have taken this. And I don't know if we really need to think about this getting an approval for a drug in this world, right? There's a lot of drugs that could be approved. IVIG, antivirals, colchicine, who knows? Like a lot of other things that could have some mechanism of action, of efficacy, antihistamines, for et cetera. But do we really need to go and recreate the wheel to say, and maybe it's important, maybe in ME/CFS we do need a drug like low-dose naltrexone, or we do need rapamycin. Maybe that's what it is. But in my opinion, frankly, if it's available, if there's a mechanism, if there's a way that clinicians can monitor response and make dose changes, et cetera, and people are going to use it off-label. I mean, I feel bad for people in Europe and stuff, okay, because like their healthcare system's different, so they can't like get access to these repurposed FDA-approved drugs. But that's something where, I think there's going to be a lot of interest in rapamycin, and but again, we've been at the front of this. David and I have, and we hope that the same groups that are doing that like see the importance of like using the same generic, stuff like that.
I don't know if they are. I mean, I know the trial you're referring to, or Aline's referring to. I don't know if how they're dealing with the product issue, the drug issue. I don't think they're doing any of the proteomics that you're doing, or we're doing, and I guess it's important to have more trials. I think I agree with what you're saying, because I think I know what you were saying, which is, do we really need an RCT, randomized placebo-controlled trial for this? I agree. I don't think we do, and that only increases the cost another tenfold. What's frustrating is they get the money, they get the funding, and it's it's hard, because I know how hard it is for us to get the funding. We were lucky; we had a great philanthropist who has supported this, along with some Cimarron dollars. But even with that support, it's 1/3 of what the other study has gotten.
Oh, at least.
And is that good use of desperately needed funds? Do they really need to do another trial like this with placebo and everything else that's costing $1 million? I just don't.
I mean, are there other options? Are there other therapies out there where that money would be used better?
That's what I'm saying.
So I agree, and I think, at the end of the day, it's there's a lot of things that we're finding out in this trial that are of critical importance. So, and we're gonna publish it soon, so it'll be out there. Cat will be out of the bag, and and again, we hope that this can be sort of a model for other things, because I've, like for example, this GLP-1 agonist, this microdosing GLP-1 agonist. I want to do that. I want to do oxygen therapy. I want to do, like, we've talked about this stuff, that we can do, and that's where the support from, key donors like this individual, but then also people supporting the work of Cimarron can be very helpful. Clinical trial money is difficult to find. Usually you're at that point, you're getting kind of an institutional level, like you'd have a CRO or other groups running it to do it properly. I think there is going to be some funding for platform trials moving up. That's what I can say from like NIH still, which is good. But those are going to be focused in the context of long COVID, which, is a thing. I do think, though, it's really important to support these sort of pilot level investigations, because they can inform something. You don't want to make a big wrong decision. Right. For example, with Rapamycin, right? I was like, we'll just get the drug from Pfizer, Rapamycin. Yeah. Pfizer's not making Rapamycin in July 2024. I was like, oh no.
But it's also, in terms of other trials, I mean, it brings up a pet peeve of mine in science, which is competition versus collaboration. Exactly. If you were another organization or institution saying, let's do a rapamycin trial, why wouldn't you contact you and say, look, we're thinking of doing a trial to validate your trial, okay? Yeah. But so what are you doing, and how can we collaborate so that we use the same parameters and save years? And it just gets me crazy that instead it's competitive and there's no sharing of knowledge to build the next iteration. It just drives me. It's such a waste.
Yeah. Yeah, I mean, when I read trials going on about things that we've been using, another doctor for years, I'm like, why? Why bother? It's sort of like you said, we already know, we've already been using it. We already know what to expect. We already know how to monitor it. We already know dosing. We've been doing this for years, and no one asks our opinion.
Right.
And I think like that comment I made was just, it was more related to like this, even the Alzheimer's drugs that came out, right, that ended up not being so great, but they were sort of pushed through, but it opened up an entire new class of drugs. So I see the value in doing that. I also think that there are other ways beyond getting a drug approved through FDA for a different indication, like getting it into up to date, right? Like getting that a statement about the use of low-dose sirolimus, that's what the algorithm
That
will
would
tell
be
these
a
docs to
big,
do.
big deal.
Absolutely.
And they'll do it
It
if
would be
they
a
have a
huge deal, you're right.
diagnostic test to order.
Plus, it also, I guess, these trials that I'm complaining about
Yeah.
further supports bigger trials, right? Because it validates certain data points, and I think that can be really useful for a larger trial, if for other purposes. So I realize there's value to everything that everyone does, but I also think that, I guess, because of at least I'll speak for myself, but I know this applies to you. Day in, day out, I just watch people suffer,
Yeah.
and I hear their stories, and they're all heartbreaking and tragic. And so I'm just so vested in trying to find the right regimen for each individual patient. And so I guess I get impatient, and then my New Yorker
100%.
comes out, and I'm just like, F these trials.
Yeah,
I'm very
I
calm.
agree.
I don't get
Yeah.
impatient. I'm calm.
Yeah, right. I'll
Yeah.
show my texts say something different.
Yes.
But I
Yeah.
did want to ask, like, just with regards to what you're learning just from your trial,
Mhm.
as you know, in specifically in the long COVID world, but ME/CFS is also considered a post-viral syndrome. And there's this controversy: is it viral persistence? Is it spike persistence? Is it T cell exhaustion? Is it reactivation of other bugs that have been dormant? Like, do you have any insight into any potential answer to any of those questions?
Yeah, I mean, I can give kind of my opinion and with some data. I think that what we really see with this sort of viral persistence thing is, we don't really know whether it's actually SARS-CoV-2 or the spike protein causing this. My sense is that it's likely causing, beta herpes virus reactivation, spanning from EBV, CMV, HHV6, 7, etc. And it just elicits that, just given that most humans, right, have these latent viruses. It can, any sort of eliciting event can kick that into overdrive. EBV-driven viral illness is well understood, in the context of this ME/CFS with relates to symptoms, but also in cancer, multiple sclerosis, et cetera, like EBV in particular has been known to drive that, obviously lymphomas, et cetera. I think that what we've found and what we hypothesize is these viruses, either in an in an initial state or in the reactivation phase, can cause disruptions in key conserved pathways like autophagy, because viruses like those are known to integrate into cells. I think that a lot of the downstream effects that we see and observe, like T cell exhaustion, et cetera, may be more related to direct changes in autophagy rather than something other than that, because again, it's an energetic pathway. At the end of the day, I think autophagy is upstream of a lot of these other findings. And, it's known that viruses can cause disruptions in things like mTOR. And if mTOR is responsible for driving autophagy, well, then you kind of have a pathway. In our study that we're going to publish here, so we can discuss the data, we found a very significant response group in patients that had documented EBV onset or reactivation.
That's we got to really underline that, because it was an
Yeah,
unexpected finding.
Did
in
not
the
expect
study.
that.
That's incredible.
There was a
actually.
clear stratification. Those patients, ME/CFS patients, everybody meets the criteria.
Yes,
Okay.
yes.
in a portion of those ME/CFS patients, there was no information on the viral etiology of their infection or the timing related to the onset of the infection, versus the other group where it was clear cut. I got mono at 19. I've never been the same. And the difference in response to rapamycin was striking. So you can say more about it.
Yeah, it was just a massive correlation, which correlations in science is like what you live for. It's like what keeps you going. But again, this was all blinded. And when we looked at the data, then it was like, okay, well, what about these other folks that don't have this viral onset? Unfortunately, with this sample size, we're a little bit weighted on the bias. We don't have enough people. But again, I really believe that the reactivation of these herpes viruses and or the eliciting events of, for example, COVID infection, because long COVID patients were included in this, those were the individuals that responded most notably. So it could be that the upregulation of autophagy, right, is enhancing, for example, T and B cells activity to clear virus. It could be that the upregulation of autophagy is helping to turn over new mitochondria that get destroyed by viral proteins and peptides that, cause that dysfunction. But again, it's the energetic source is what we're interested in, and that viral onset type it's very plausible that the
Mhm.
viruses can cause that.
That's fascinating.
Yeah, what you just said got me going here. Wait a minute. So I think what you're saying, and it's really important, is it's not just that rapamycin upregulates autophagy, which cleans up stuff. It's just a waste management system. It's by virtue of that upregulation and waste management, it improves the functionality of the immune system and key immune cells, which may then address the viral reactivation and the other crap that's going on. That's really important.
That's huge.
And it's
Yeah.
important to
Huge.
know too that autophagy goes on in every cell of the body. So
Right.
neurons, for example, that are able to fire, neurons that fire together, wire together. So you think about things in the context of brain fog.
Mm-hmm.
We had a really interesting small group, very small, but of this adverse events or events that people report to me. And a couple of the patients, not a couple, but a significant portion at least, talked about this transient insomnia that they got.
Mm-hmm.
So they would take the drug and then they're like, I just feel jacked up. And I was talking to the patients and I said, okay, like feeling like just trying to figure out what they were talking about. And it sounded to me like a couple of patients was like, well, I was just able to read all night. And I was like,
Read,
ooh.
you said?
Yeah, I was like, is that your brain waking up?
Yeah.
Is that, is the
It's
process,
possible.
so,
Yeah.
and that's again where we know that this process of autophagy driven through mTOR, that is in every nucleated cell in the body. So skeletal muscle, immune cells, APCs, like neurons, and understanding how those even slight changes, even if you have like a 3% decrease in autophagy function, it can cause so much of an issue throughout the entire
Yeah.
body because it's everywhere.
Yeah.
And did you also include? I mean, I'm assuming they were mostly your patients. Or?
No, I don't. It's not mostly mine.
Yeah,
Is
we've got 6 other clinicians.
I'm just wondering, in addition to viruses, did you also have any kind of evaluation of tick-borne diseases?
it was not part of the inclusion or exclusion criteria. So, I mean, it's a great question. As you know, and people listening to me know, I'm convinced that the tick-borne infections are exactly like the herpes family infections.
Yeah.
They are reactivated diseases. So I was going to say something else about the biobanking, but I'll use it for this as well. The fact that you have these samples means we can go back and say, well, these 37 patients didn't respond. Why? And we can run, tick-borne studies on the, infection studies, and we can run heavy metal studies, and we look for other causes other than the initial infection cause for a non-responder to see if there's to figure it out.
No,
and,
exactly.
And certainly, you're right. Tick-borne infections could be it.
Yeah,
And
absolutely.
we've been really lucky. Like Cimarron, we have our good collaborator Connie Knox, who runs Copa Laboratories based up there in Waukesha, and that's been her entire career, has been tick-borne, arthropod-borne infectious diseases, and so that's a plan of ours. I think because we had taken kind of all comers, this is sort of a post hoc blinded analysis. But I agree. I think, but it's the same thing with SARS-CoV-2. Like, do I think it's possible that that's reactivating, causing it? I don't, but I think it could be an initiating response to upregulate, herpes virus reactivation, for example.
Yeah.
And,
now that I realize it, I should look at my patient list because I probably have tick-borne infection lab results on almost every patient I send you because I look on almost every patient.
Yes.
And we didn't include that in data collection.
But even if it's older,
we can look back and see.
Yeah.
Yeah. It would be interesting because there have been some tick-borne disease evaluations in other long COVID trials, and not necessarily about rapamycin, but just in other kinds of trials. And so, I'd be curious.
Yeah, and I think I kind of in my brain I've always put these patients into pretty simplistic terms, but like one phenotype being this classic viral onset beta herpes virus reactivation world, and then this other slower, gradual, more autoimmune-ish type subset, which again, it's just a different thing. The symptoms are very much the same, but again, the mechanisms and how people respond is maybe dependent on some of that initiating factors. So it's it's a lot to look at.
I know.
Yeah, it's fun.
I know. There's so many things. There's so many variables, right? And that's what we
Yeah.
talk about with the heterogeneity of this disease. There's
Well, it's conceivable that those variables in the responder, non-responder, what you just said, that the solution may lie simply in what we talked about earlier, which is dosing.
Yes.
The non-responders may have just
be
need a
that
higher
simple.
dose because they weren't viral mediated. They were autoimmune mediated. We know
Right.
rapamycin can treat autoimmune disease, but
Yes.
maybe you need a higher dose.
Right. Yeah.
Hmm.
Yeah.
Maybe I should start increasing the dosing.
I've done it a little. I get nervous still. Maybe when we publish and we have more,
Yeah.
I'll be a little bit more bulletproof.
No, and that's where I just also am so thankful to the patients for
Yeah.
participating
Oh, yeah.
because honestly, without them, none of this happens, and they've been just incredible humans. And I can say I've gotten to know some of your patients over this trial, and they're just all wonderful,
Yeah, they
wonderful
are.
people.
They really are.
So
So when does the paper
I
come
was just
out?
going to say, all right,
Yeah,
And
let's
in what journal? Where am I looking?
let's
yeah, we're gonna
unveil here. a
yeah.
little.
So we've got an initial draft that we're gonna be circulating for the submission here next couple of days. It's going to be submitted at least initially. I'm not going to hold off on saying the journal name just because, there's embargoes and stuff like that.
Yeah.
A very good journal. A very good journal. Just kidding. No, it'll be a good journal. And but like I said, nowadays what they have the option for are these called sneak peeks, so you can see the preprint online. Before this, you used to have to wait like an embargo it for like 10 months before the peer review process went through.
Yeah.
But as it's under review, people will be able to see it, comment. I think that'll be important for us to be like, hey, we did something, Avik and I, and kind of when everything's going wrong in the lab and nothing's working, it's like we always get to remind ourselves now, like, hey, like we tried something, so it may not be perfect. It will be what it's going to be. But yeah, we're excited. So in the next 3 weeks is when you'll start seeing it online.
Oh, wow.
So maybe we can share some preliminary results. We won't,
definitely.
This episode won't be posted till after the publication or the preprint.
So yeah, I can summarize the findings. We had about 40 patients complete the entire baseline to T3 program. We had a number of patients that were initially consented but didn't start the drug. We had a series of patients that either failed the ramp period or failed the different time points. Interestingly, though, it really had nothing to do with adverse events. It had to do with the study design.
Mm-hmm.
We didn't have funding for phase 1 to pay for things like the study drug and the labs, which actually got expensive. We also, just because we were not sure how this was going to go, the clinicians, they really, I think, and David can talk about this more, we were really listening to people. So if folks said they weren't feeling better, they just stopped it. Because, why would you keep doing it if you're not feeling better? So that's
at what point did they stop it? During the titration or when they were at the
each time?
dose?
Most of it occurred during the titration time point. We had obviously fewer and fewer stoppages by the time we got to T1, T2, T3.
Yeah.
But that's one of the reasons why. And people are going to say like, oh, well, you started 86 people on the drug. Well, you only got 40 that went all the way through. Well, it wasn't because we had a 50% massive AE rate. And we're being very clear that yes, there were some AEs, but it was not related to the stoppages. It was more functional items that related to those stoppages. So anyway, of the 40 people, we saw a significant response. Over 80% of these people responded beautifully.
Response in what specifically?
By measures of the multidimensional fatigue inventory, the short form 36, the Bell activity scale, and then the specific symptom inventory.
Excellent.
Those were our key time points.
And to talk of science, as you say, there was a correlation between those improvements and the changes in Beclin and ATG13.
Mm-hmm.
That's cool.
So basically, it's like when we're writing these papers, normally it's like as basic scientists, Avik and I were talking about this the other day, it's like, well, there's all these images of cell pictures and all these other things. It's like in a clinical trial paper.
Yeah. It's
like you got a table and you got a couple of graphs, and man, that's the most important piece. So the correlation between Beclin, like David had said, that we saw a significant change, positive changes with the questionnaires correlating with upregulation of Beclin, and then the same correlation with the downregulation of ATG13. The viral onset patient population was the most clear group of people who are likely to respond. So you could even consider thinking, hey, well, maybe an inclusion criteria for the next phase of double-blind and randomized could also be make sure the person has a viral onset.
Mm-hmm.
So the other way.
Yeah.
We did a third correlation, which is looking at individual responder status. And that's where very recently we've kind of developed this machine learning model to compare how the questionnaires, the questionnaires are limited. They have floor and ceiling. There's like receiver operator bias. There's also just bias on how many times these patients fill out the questionnaire. So there's kind of limits with that. But at the end of the day, tracking the responders and then seeing, okay, this person said they went from for example, a 0 on the Bell activity scale to a 40 or a 50, which is double the improvement. We see a direct correlation with their Beclin levels changing at, 30 to 40% folds. I mean, these are massive changes to the level. What's nice with Beclin is you can compare it to like elite athletes. You can compare it to group controls that are out there. At the end of the day, the drug was very safe, well tolerated. This go around, this next phase with Ageless RX, and the next phase of this, we just need some more infrastructure,
Right.
And I think we're just going to keep adding to it. But this is the first ever biomarker directed clinical trial in ME/CFS where we have a mechanism. I'm really proud of that, and I'm so thankful.
You should be.
So thankful to you guys. So
no, it's a landmark study, and no matter how small or pilot it is. Honestly, I think that this is exactly what the ME/CFS patients need.
Yeah, and just to, on the size, it's like people forget it's like 4 time points, with 40 patients is actually a lot.
It is a lot.
And that's just where, building the biobank is going to open up. So we have about 4 papers that are going to come out with this. So we're going to publish this first one as the kind of like the protocol paper, but then for the next 6 to 8 months, you're going to see 4 additional publications looking at all these cool things that are coming out. We're going to be using Seahorse to look at PBMCs. We're going to be looking at changes in immune cell profiling. We're doing a lot of flow cytometry. Ooh. We've got a wonderful Shimadzu core metabolomics, proteomics at medical college at University of Wisconsin Milwaukee that we can get on. We have NMR. We have all of these sort of core facilities that now that we have these samples and we have wonderful graduate students and all this is being led by Avik, myself, we have projects for people. And so that's going to really drive our work. It's going to drive our next grant cycles. And I think that we're really going to be able to find some key mechanistic findings from this study.
Immunoprofiling would be so interesting of these patients.
We've shown, again, you can do this work, like we have the ability to do it, and that's something that we're just so interested in, because again, the shifts of what happens with rapamycin, one of the downstream effects we hypothesize is this shift from this M1 sort of pro-inflammatory phenotype to an M2 in macrophages, more inflammation resolving, greater phagocytosis, like greater removal of things, reducing that type of inflammation. Then when you do a cytokine readout, it could maybe correlate with that. But at the end of the day, those are all options. And Seahorse, like you got to be able to look at the changes in glycolysis, oxidative phosphorylation, all of those. These are all hypotheses that we can test on these samples. So
yeah, I mean, Seahorse, that would let you really link the change to mitochondrial health and disease. So in phase 2, how many patients are we aiming to enroll?
About 100.
Okay.
Yep.
And time points are still stop at T3, or are we going out? I know the answers. I'm telling you.
Yeah, we're gonna try to get people to focus getting through baseline to T3 first, and then we're gonna, that's when we're doing a monthly time collection instead of. But then after T3, it's every 4 months.
Okay, good.
We can do things like we can ask interesting questions, like maybe people should be on a holiday, or we might have enough patients that might want to stop or take a break. And again, that'll just be, we're going to want to publish that quickly. So we'll have probably a cohort that gets through baseline to T3, and then we'll have another cohort that continues on further. The sirolimus quant testing and sort of this individual pharmacodynamics study that we're doing, that's going to happen in between T2 and T3. So the patient will be established on the drug. The samples, we've increased our ability, like I said, with these PBMCs to get them. So that's really important. The last thing you want to do is spend a lot of time doing Seahorse or something like that and then realize oh, all the cells are dead. It's a mess. So again, that's where we're just going to keep going we'll run this until the wheels fall off. That's kind of how I feel about it, because, this is the future of the lab, Cimarron. It's the future of our grants. We have a lot of other desires and wishes. Like I said, one of the benefits of working with a group like Ageless RX is that it kind of lets the doctors focus more on, I think at least, selecting the patients and doing stuff without having to be like, oh, I need to bother Dr. Kaufman for like a lab rec form or something, or like
refills.
a refill.
Refills, right.
Yeah, and they do that, and that's something too where they have a lot of other therapies like GLP-1 agonists. They have NAD+.
Right, I
They
think,
have
yeah.
things that are like,
So there's a benefit to them because they offer a lot of other drugs. So.
and then doing a good job for them. Hopefully they'll like continue to want to work with us.
Right.
And that's kind of the goal.
So you are enrolling new patients for the second phase.
Yep, we are. Yep.
And so if they've already been on sirolimus, is that an exclusion criteria?
No. We have a couple of patients that have been established, and one of the clinics, Dr. Jen Bell at Bateman Horn Center, we got over this question the other day. I said, well, why don't you just have them stop the drug for a month and then redo the ramp?
Okay.
Because it is a different compounded generic than what they were on before. There's also an option for people if they want to skip the ramp and just go right into the main dose. They can. That's an option as well. I'm trying to just make it uniform because we have kind of smaller numbers, so any sort of changes in the protocols makes it difficult. But yeah, if someone has been on it before, we'd love to have them, and try it again. Okay. And so, the IRB is very flexible. Like one thing that the deal with Ageless is, they trust our clinicians. They know that you guys are all watching these people. So like, that's why they're willing to take on some of the liability with prescribing, et cetera.
They're more conservative than us.
They're more conservative.
That's been one of the issues. There's a learning curve here because they'll look at my patients and they'll say, well, I don't know if we can take that patient. They're on 9 drugs and they got tick-borne infection.
Because aren't they just like a mail order?
Yeah, but they're actually, they have doctors they have a questionnaire and everything, but they're conservative because it's mail order. So one of the challenges was the under 40 female entry was an issue because they didn't want to take anyone who could get pregnant.
Oh.
Because they have liability issues, but it's worked out. They've been a total pleasure to work with.
Yeah, and I think that's been very relieving to me because, and they've done this Pearl anti-aging study. That's a very kind of seminal piece of work in there. So they have Stephanie Morgan, who's their chief scientific officer. She's really smart, and again, if, for example, if we got funding to do a double-blinded, randomized, placebo-controlled trial, we'd have a mechanism to do that because they have the placebo. I mean, there's these things that make it just more functionally easy.
Well, also what you said before, that having this study model, these decentralized models set up and linked, partnered with Ageless, overnight, to some degree, we could do the same study with GLP-1, the same study with acarbose, the same study with metformin. Again, all these drugs that we think have benefit, but we don't actually have mechanistic proof of that.
Right.
So,
like the next one I want to do is metformin with Rapa. I mean, I think that's some of these people. I
regularly prescribe them both.
Yeah, because and that's
together.
The
I
thing.
mean,
and that's just something where even in this current phase 2, the IRB doesn't care. They're like, well, you're using FDA-approved drugs, so it doesn't matter.
Right.
So you can do whatever you want.
No, exactly.
Because what we're really doing is we're just watching you guys practice medicine. That's it.
I don't know if everyone would say that we practice medicine.
That's what
sorcery,
they call it.
Sorcery, yeah.
Sorcery.
So it may not apply directly to our patients, but I'd love to see the exact same study model for people over 65 where this all started, so to speak.
Yes.
And see, I mean, what is my Beclin and my ATG, right? Before I started my Rapamycin and now that I'm on it, there's tons of data on anecdotal, as we began in the beginning of this talk, with Rapamycin and the Manic study and everything else, but nobody's done anything in the geroscience world like this with looking at the lab benchwork to validate it.
And I think that's where the publishing this with getting that, especially the ATG 13, that's a brand new thing we built. So that's gonna hopefully get a lot of folks interested. Maybe even Dr. Manic's group would be willing to send us samples, because a lot of that can be done on banked stuff too.
Mm-hmm. Yeah.
So
that'd be an exciting trial.
This is really exciting. This is exciting stuff. I'm taking Rapamycin also. We're all taking it.
Yeah. No, I mean, it's, we think there's a lot of implications for this. One of the, in our animal model, had one other research. I didn't believe her, but ATG13 knockdown animals are nude. They're hairless, no hair. The second that we gave rapamycin to that animal, it started growing hair.
Stop.
So I kind of think from I'm kind of
No, it's not working for me.
I don't know what there is, but we've been very interested in this mechanism, obviously in this patient population, but sometimes in science, you fall into things, and that can also help with funding and things like that. So we've got a mechanism now looking at bladder tissue and how that works with aging and with the help of rapamycin therapy, but drilling down on that mechanism, and hair follicle growth is driven by autophagy. So there's a lot of potential, and that's why we're just excited to do it. So,
well, it's great. It's been really great.
Yeah, we learned a lot today. Thank you so much for coming on.
Thank you. Thanks for having me. This is my first podcast. So
really,
yeah. So
wow.
You did good.
Thanks.
Yeah, you did
Appreciate
great.
it.
You did great. You explained things very well, and I think you got all of our listeners very excited over rapamycin.
Yeah, and
I think it's great to be able to discuss the real science for our listeners and see the stuff we're talking about in more, biochemistry detail.
Definitely.
So that's great.
Definitely.
Really good.
Thank you so much. And all of this work can be, you can look at Cimarron.com.
Yeah.
It's all there. I think we've published 6 original research articles this year alone, so we've been productive, but this is the next phase. So thank you so much.
So
again.
do you want to sort of do a shout out to tell our listeners what you need?
Oh, well, we need money.
Exactly.
There you go.
We're a 501c3 nonprofit.
Right. Those are
Every
important
every
details.
research dollar goes directly to projects. You can name a project. You can donate $1, and I'll use that dollar exactly for that project. And we just really appreciate it.
Yeah. Well, we appreciate you and the work that you do.
Thank you.
Obviously, I mean, that's why we have you on, because we want to help. We want to help everyone.
It's been great for me because as a clinician, I rarely get to work with a scientist like this. Mostly, even back in the HIV days when I did all those studies, I never spoke to anyone in the back office.
Yeah, you didn't interact with the scientists.
They usually keep us in the dark.
Yeah. So this is another sort of unique aspect of this trial.
Totally.
Where most of the time it's either basic science or clinical people, clinicians. Rarely do they work together. So you and I are texting or talking or emailing several times a week.
Yeah.
Meet.
What?
Yeah.
What's that term? Ne'er will the two shall meet.
Yeah, well, we're changing the rules.
Changing the rules.
We should change the rules.
Yeah.
Because that's what we need to do. We need to take what they're learning in the lab and we need to bring it to the clinic. And I know that's what translational medicine is all about. And, but it's not done effectively or efficiently. So we need.
And we can't do anything unless we know what the hell we're looking at. So,
right. Exactly.
And
I think patients need to also play a bigger role in research.
Yeah.
And a lot of the patient-led collaboratives have actually done a lot of
they've been game-changing.
Yeah.
Yeah.
So it requires all of our perspectives, the patients, the clinicians, and the scientists.
Okay. Well, thank you very much.
Thank
It's
you,
been
guys.
a long,
Appreciate it.
I think we almost talked 2 hours here.
Wow.
Yeah, we're
All
almost,
right.
Yeah,
Pretty amazing. Didn't feel like 2
Yeah.
hours.
No,
but,
not at all, actually.
This is great.
All
I
right,
could probably
fantastic.
keep talking, but we'll stop.
Yeah. How long are you in Seattle for?
The weekend.
Oh.
Yeah. My wife's getting in this afternoon, so we're gonna go look around. She's got a meeting with the group over at UW, so
Fantastic.
who knows? It's pretty nice out here. I thought it rained a lot in Seattle.
Yeah,
I was
it's been
like,
so beautiful.
Yeah. We lie about how much rain.
that
All right.
makes
Well,
sense.
thank you very, very much. I hope everybody enjoys this. It'll probably be posted about 4 weeks from now, I think, right? So please watch us. Please subscribe. Do that YouTube subscribe button. Apparently it matters. And send us your questions and comments and suggestions for other episodes. Thanks very much.
Bye.